Okay, good morning. So I would like to start with a couple of words about this homework. So the homework was about a paper. So this is a paper I want you to read. This is not a very, how to say, the high impact paper, but the methodology I want you to be clear. So this is I want you to answer a couple of questions either by writing an essay or speak in a video. So a lot of you say, okay, this is a random forest. That's right. So the algorithm random forest is one of the classification algorithm. So basic task of this to do a classification. Then my second question was this technique was used in its native way, or it's not, because they are not classified cells into patient or control. They know the labels of those cells. They use this algorithm in the opposite way. So they know the answer, but they try to see, okay, which cell type will give you the best prediction of disease and healthy control cell to separate them. So if you have a cell type, can separate the two states of cells 100% accurate, that means this disease probably strongly associated, caused by the change of this particular cell type rather than other cell types. So they use this algorithm, is a classification algorithm, but used in the reverse opposite way. They know the answer, say which cell type gave me the best answer, then they prioritize this particular cell type. See, okay, this cell type will be deserve further investigation because they can separate cell states if you know the states of cells. So if you can somehow answer in that direction, you will get a better score in this task. Overall, this will be the second last homework. Then we will have another homework. In total, we have 4 homeworks. So this is like this part of the final exam, and then you have platform presentation by the end of the 13 weeks. Each of you will spend like less than 10 minutes come to here and discuss a project, discuss a paper, or whatever you learned, reflect the knowledge you learned from this class. Then everyone will receive a score sheet. Okay, they will vote, say, okay, there are 4 categories of the judge of this performance. So one is significance, right? So everybody will receive a score sheet based on the significance of your topic you talk about. Then innovation. So whether in your talk there's some innovative, creative thinking in there, and then approach. So whether you gave some method development, or how are you going to achieve if you define some questions about your studies. So it could be a proposal. Then at the end will be your performance. So how you communicate with people during the platform talk. So each one will have your name, 1, 2, and after that you will rank those speakers, including yourself. Say, student number 1 have a significant, rank number 5 after you listen to all the students. And the second student, in terms of the significance of the study, 1, so you put the rank here. And then in terms of innovation, this could be 1, this could be 2, 4, after that. So I will collect all this score sheet afterwards, and based on the rank, I will give you a score, okay? So if you performed number 1 here, number 2 here, number 1 here, the student number 2 will really have a high score for this part of the test. And then you have final exam that will be 30 multi-option, multi-choice. This exam that you answer is just 4 answers. You take one of the correct answer, and then so you have homework and presentation, and the final exam, and each is just 1/3 of the overall score, and then I will at the end summarize them. Is that clear for students? Okay, 3 parts. So probably it's a good time for you to start to consider what are you going to talk in your 10 minutes, including the answer of the questions. So you probably want to finish at 7 minutes, and then spend another 2 or 3 minutes to have a question communicate with the students, audience. So that's your presentation. Okay, today we are going to talk about, question for the.
Sorry to interrupt you. Are you going to post the details about the project on Teams?
The project required the score.
Right.
I will put that information in there so you know what type of project you can discuss. Okay, so components and all decomposition. So this is outline for today. So we will go through matrix decomposition and also several different methods, NF, and eigen decomposition, SVD, ICA. Okay, 3, all the decomposition methods. So this is a basic task for data scientists. There are many, many different ways to do the matrix decomposition. So this is a list from Wikipedia, and we will go through some major ones, okay? So this is from a very beginning. I remember this is like second lecture. We talk about matrix multiplication. And you have 2 matrices you put in this way. So this will be 1 times 7, 2 times 9. You compose and put the number here. So that's an example, you remember. That's you and your roommates, and want to make 3 sandwich. And the 3 sandwich contains 6, each contains 6 ingredients. At the end, you have this matrix. That's you and your roommate. Each of you will buy such amount of the different ingredients. Okay. So at that time, I remembered I mentioned if I gave you the product of A and B, if you have this result here, please consider whether you can reconstruct A and B, the component. In this case, it's almost impossible because there are so many different ways. And even though I tell you there are 3 sandwiches, it still be very difficult. This is because the sample size is not enough. If you do that again, give you different number combinations, in this right now is you have a 4, 5, 3, but if you change the number, give you enough those samples, it's possible. And also, those components have a non-zero property, and also the price or something like that. There's a boundary, and you know it's non-negative. It cannot be over 1,000 or infinity. So there's some possibilities, and you can actually figure out what those numbers, what each component, each of the sandwich will take. Okay, if you gave you enough samples. This problem, if you translate this into single cell, we have to understand each cell is not a single function. Each cell may have involved in multiple functions. Each function is also associated with multiple genes. So if you consider the functions that each individual cells get involved in, we name them like gene expression program. And each cell have multiple programs to play with, okay? So let me just draw a matrix, and then we look at the decomposition part to give you the whole framework. Then we go into details how to do that later. So as I said, we have X matrix, right? You know this is cells, cell 2, cell N, then you have a gene 1, 2, gene N. That's what we measured. Okay, we use shotgun sequence to sequence those mRNAs and count how many, for this particular cell, how many rates located in each of the gene, and then another, because of barcoding system, we have this matrix. It's a huge matrix. But if you consider this particular cell, say cell 1, then if you know cell 1 get involved in a couple of different programs. In this small example, I will see cell 1 only get involved in 2 programs. So we have a program P1, P2. So cell 1, cell 2, cell N. So the P, we don't know how many P. So here we just assume K is 2. So this is called latent variable or latent feature. We have latent variable P, that's cell program. And the number in here, for instance, this will be A1, B1, A2, B2. So those are just some numbers. It can be integer, can be just a fraction. So you see how much or magnitude or levels of each cell get involved in P1, how much get involved in P2. It can be normalized, say 50-50, 70-30, whatever. Whatever numbers, there's a scaling factor that you know. Okay, on this side, you have P1, P2. And you have a name of genes. Gene 1, gene 2, gene M. You also have numbers in here. Okay, maybe C1, D1, C2, D2, It's just integers or some scaling factors. You see, okay, gene 1 involved in program 1, program 2 by 50-50. Half of the transcript involved in this. Gene 2 maybe 0 involved in P1, 1, 100% involved in P2. So it depends on your numbers here. So you have this decomposition factors, those numbers here. Okay, you should consider P1, P2 as a sandwich here. Here we make 3 different sandwiches, but here cell gets involved in 2 programs. You don't know this, but you just assume you know you have this information from the database or gene set collections. Okay, if you have a gene set collection, say gene 1, 2, 3 involved in cell cycle, and the gene 4, 5, 6 involved in apoptosis. So the different program names. Yeah, Luis, you have a name question? Yeah.
Just to be clear,
yeah.
that P are given values or are calculated by the user?
The P will be something we are interested in. We don't know.
Okay.
Yeah.
We predict or we get.
Yeah. So if you have half of this matrix, you have this for sure measured from experiment. If you have a part of knowledge of this, you can compute that. If you have part knowledge of this, you can compute that. So everything. But if you don't have any of this red and blue matrix information, you have to estimate completely from big X. That will be much difficult, but doable.
But that estimate values are in the time 0 or is like a prediction?
Which value?
P, for example.
This one here. Yeah, so those are just, you can estimate.
Okay.
Yeah.
And that number represents the involvement.
The involvement, the strength of involvement, the committed, yeah. Yeah, so this is the idea. We try to decompose this end product here. So this is, the R there is this X. We try to estimate U and V, okay? U is this, V is that. So this example is Netflix, maybe, the users and their movie and the rating, okay? If you have this matrix, you want to see, okay, how many different categories of users, okay? And then each user, what their favorite movie score. In this way, you learn the latent variable, the D, and how many different categories of viewers of those movies. The product of this will be approximate of this rating sheet score. Okay, is that clear? So this procedure is called non-negative matrix factorization. Or if you don't put a non-negative, the matrix factorization is just global. There's different ways you can decompose this into the product of the 2 small ones. But in this non-negative matrix multiplication, there's requirement that all the values in these 2 matrices are non-negative. So non-negative means 0 or positive values. So this is very useful for us because we don't need negative values in this. Let's call this U and UV. So this U This is a V matrix. There are so many different matrix factorization or matrix decomposition algorithms. This one is relatively new. Okay, it's the most effective way to compute. The algorithm was invented in 1990s, maybe. Yeah, so that's here, 1990s. So this, compared to others like SVD or other algorithms of decomposition, this is relatively new because the algorithm require all the U and the V elements in these two matrices are non-zero. So this gave you some of these restrictions of you can predict those values. Those restrictions actually make the algorithm more difficult to achieve. So that's why this computationally efficient algorithm was developed until 2000, even almost 2000 year, 2000. How this was done, okay? We don't have to go to too much details, but the general idea is this. So you put some random numbers, A, B here, and random number here, and you make a product. It's very easy to compute the product of two matrices. And then you compare with your observation, X. So in this case, X is our V here. And you compare the difference between UV, okay? So you have U times V. This gave you a prediction of X. Then you use X minus it. You compare X with that. And you take a norm for this whole matrix. You want to minimize this difference. The smaller this difference, the better prediction you make from these two matrices to the original observation. Right? So that's your function. You want to minimize this function to make this product W and H as close to the V as possible. The notation changed a little bit. We UV and different. There also another algorithm similar, same idea. The cost function just defined a little bit differently. Here they use KL divergence, okay, computed based on the log of this ratio times V minus this. This was introduced by Li and Xiong in 1999. So based on that, they construct or invented this algorithm through the iteration of a loop and to reevaluate values. So you start from the random number here. After that, you change those numbers based on this result. If it's good, you will change a little bit. If it's bad, you change drastically. So through the loop, so you start random, then compute this, and come back to reset the values here based on your product here, and then do that again. After several loops, the numbers of those A, B, C, D will not change anymore. Then you reached a state called convergence. So you convergence, so after the iteration, then you reach to the conclusion, this is the best solution I can get from this algorithm. So the equation here, I will copy that to the next page, is to show you how to update those values based on the difference you detect between prediction and observed. So here is, you can either minimize this directly or follow this definition to minimize this divergence. It's the same idea. Minimize this or that, two different ways. This is the way to update. So that's your initial guess of your values in here. Random values you start maybe. And then you update those values based on this result. And you keep updating. You update this and that in the same iteration. And do that again, repeat. Yeah, so this is the explanation of why you can do that. If you decide to minimize this, you will have to open the expression right into this way. This is the same idea. Then you get this loss function, okay, from those derivations. Then you take the differentiation and then to compute the derivative of those functions and try to make it equal to 0 to allow you to reach that optimal solution, so that at the end give you the way how to update the values in the U and V. Again, this is a repeat, same thing. Okay, to update your W and H, which is in those two matrices, and then to iterate. And this is one of the solutions. So if you want to approximate this matrix A, you can decompose it into these two matrices. So that's inside the source code of those implementation. So this implementation is in MATLAB. If you open this implementation, you can see there are options. If you want to use Euclidean difference, then you use this equation in order to construct your cost function. If you want to use KL divergence, then you construct it in this way. So you can see this is very close to the mathematical expression you see from the previous slide. So that's the difference between this and V hat is products of U and V. And then this is the difference, it's the norm of this. Okay. If you use divergence measurement, then you have this log, this ratio, and basically this copy of the mathematical expression. And in this expression here, another implementation, I want to show you the update. Okay, I tell you there's iteration until you reach to the convergence. So this is matrix multiplication and divide by this, and then you update the values in H, and then you update values in W. Then do that again. After several rounds, you reach the iteration after iteration. So this is MATLAB NNMF function. So this is an official release from the package of the statistic toolbox. So what do you need to give this function is the A matrix. And here is our X matrix. You also need to give the number K. Okay? So what's the K here? The K is 2. The sandwich example is K equals 3. So you have to give the algorithm how many different components you think will be involved in X. Okay. So I want to show you different ways to write this. If you so now this X can be written into X can be written into the U and V. Okay. So in our case, is this U is n by 2. The V is 2 by m. So X will be n by m. So this is dimension, right? So we write in this matrix multiplication way. You can also write that into X can be also write into C P1. So C1 Cn P1. Okay. So this times P1 G1 Gm. So I'm writing not a matrix, but it's just one column here and one row here. So this is just one program, a vector times another vector. But it's not the inner product, it's outer product. So this will create a matrix. So X, say P1. So this matrix is the same size of the original. And this will be plus, again, another matrix, 2 vectors multiplication. In this case, it will be P2, P2, still C1, C2, Cn, G1, G2, Gm. Okay, instead of writing, so let's write this. So instead of writing into 2 matrices multiplication, I write that into 2 vectors multiplication plus another 2 vectors multiplication. Why I can do that? Because, let's see, this is A1, B1, C1, D1. So the first corner here is A1 times C1 plus B1 times D1. So the value here, if you compute, is in this way. Same thing here, A1, C1, B1, D1. So the corner here is A1 times C1. Corner here is B1 times D1. So this one, A1, C1 plus B1, D1. So the value here is the same as this one, right? So you just write in the as a matrix multiplication or vector outer product, multiple vector outer products, right? Same expression, but write different way. Why I want to write it in this way? Because if you realize P2 is not important, maybe just very small portion or very low expression give you values. So a lot of values here will be close to 0. In that case, you can delete the P2 altogether. You only use P1 to approximate the observed, because P1 contribute, say, 98% of all the expression of your observed Px. And this is only less than 2%, that you can delete the whole program. If you think this is a noise, you can remove this from the data. Depends on your estimation of how much all these values are in there in each. But this will be difficult to look at the contribution of each, because you write everything together, they all summarize together, and there are two different layers. P1, P2, they put together already. And this one helps you to separate. Put in the different terms, then you can decide where I should stop. I probably only use top 2 if you have 25 programs. And only top 3 will be enough. I delete all the rest of them. Okay, so this is a Python version, and to write an MF in a package called syskit learn. So this idea had been used even before single cell data become available. So this is the old PNS paper, 2004. They defined the latent variable K gene expression program. They use a name called metagene. So metagene means a program, basically. Okay? If you have a gene 1, 2, 3, 4, and several, and involving this, so they try to see, okay, those are the metagenes, and all encoded for program 1. So just different name, but then they called molecular patterns discovery. Again, that's just different names, molecular patterns or metagene. So this could be molecular patterns. So they want to learn, decompose this observed gene expression matrix into K different metagenes, and each metagenes among your sample have a different expression or molecular pattern. They try to discover how many metagenes in this matrix, how much each of your samples getting involved in different meta-gene encoded function program. So this matrix was, they have samples, okay? Samples they get from microarray or RNA sequence, and each is from one sample. It's not in one cell, okay? At that time, they don't have those. In this example, they have two programs, okay? Only two columns here, and two rows on the top. And that's values estimated from each of the samples. So program metagene 1 is this blue, okay? And metagene 2 is this green. So you can see the samples have been automatically, based on the pattern of the magnitude of those metagene program involved in, automatically separated into two classes. Two classes, 1 and 2, and in one, program 1 have higher expression, where in class 2, the program 2 have high expression. But if you only look at this, you'll only be able to tell the difference, okay? You have to decompose this, and clearly those patterns start to show, okay? So those red indicate a high expression, high value, high magnitude, and those are the blue are low. So you clearly separate samples, columns into two categories. And those are the more recent review paper talk about non-negative factorization applications in computational biology. Okay, many of them. Yeah. You mean this matrix here? P1. No, each gene may have multiple functions, maybe involved in P1 and P2 at the same time. This one here, you still have numbers. If you have a B, D, those are non-negative. You still have, so cell 1 involved in P2, how much? And P2, gene 1 involved in P2, how much? So you still have values. If you put zeros here, then that tells you there's no contribution for P2 project involved in a particular gene or in any of the cells. They still have values. P1 is not a cell. P1 is each cell, how much they involved in the program 1. It's a value, yes. Yeah, maybe we just look at those different terminologies may help you. So again, same idea, okay? Sample data, the big X, to decompose into, this is called factors, okay? Now instead of programs, it's a more general term, factors. How many factors involved in this? And then you have different genes and then factors involved, and then you have factors, and for the samples, how much. So you have amplitude for gene and the sample factor patterns. So they're just named differently, but the same idea. So either you consider this as a gene factor, sample, or sample factor gene, doesn't matter. So whatever, they just decompose that into this. But you have to know how many factors in there. You have to predefine the factors. Same here, right? Gene factors, factor samples. Samples you replace as the cells. We have individual cells, and how many different factors each cell get involved in. Sample relationship and the molecular relationship. This is one of the examples of applications of non-negative factorization to understand the different cell type involved in. So you can consider this as a cell type. So this example is not for a single cell. So this example is from a whole tissue, sample 1, sample 2, and then you have you separated the samples based on the genes expression in different cell type. Okay, so you have this matrix here. It's a gene by cell type. So consider this matrix C here as here. Okay, you have a gene by cell type 1, cell type 2. And here you replace cells with samples. It's a whole tissue sample. There are different cell types. Then you sample 1, have a cell type 1, 50%. Cell type B, 50%. You have 50-50. If you have a sample 2 only from cell type 1, then you have a 100 here, 0 here. Okay, so you can use this matrix. If you know this matrix, you can use this to basically you have half of this matrix. You have observed whole tissue samples. Then you can compute for that particular sample, cell type 1 how much, cell type 2 how much, the percentage of cell types in the sample. I think they defined cell types that get involved in cancer, and then they can see the fraction, the proportion change from the healthy sample compared to the tumor patient sample. CyberSort is a commonly used package to do cell type decomposition, or they call it deconvolution. So different names, it's just decomposition. In this case, you need to have different cell types, a database, and also which gene they express. So you have this blue matrix information already. And you have your observed sample and the gene, so you have this observed expression matrix. So once you have this two, you can easily compute this one, okay? Because you know this times that created this. So you can compute from this by giving database from the cell type. Then you compute for each sample how much of each cell type in that particular sample. If you have a sample, say, all from one cell type, then you only contribute all the genes from this cell type to create those. Now, if you have a sample, have a different cell types, each of this will have some non-zero values. You know, the percentage of a different cell type composition of your sample. So here the sample is a bulk RNA-seq sample. So CyberSort is an algorithm applied to traditional bulk RNA-seq multiple samples, and using information from a single cell type marker gene database to predict or estimate the relative fraction of each cell type in that particular sample. So in this case, for example, they say, okay, in your whole tissue sample, you have this percentage of the blue cells, this percentage of orange, okay? So gave you the percentage of the composition of your sample. The procedure is called deconvolution. Whatever you call it, it's a matrix decomposition algorithm. This comparison between two. This package is extension of the CyberSort, but it utilizes the information from single-cell RNA sequencing data. Okay, so this is 2016, and the original was a little bit earlier. So you need to give it input, different cell type, alpha, beta, gamma, data. Then the gene, marker genes, to give the information. So this is like a marker gene database. So this is like this one here. So you know you are not completely dissociated this. You know part of the information here. So you can do a better job to predict the values in this matrix. This is another one said it could be more tolerant to just random stochasticity. Yeah, same idea here. This is very useful nowadays, even more so because this spatial transcriptome becomes so popular right now. However, this method is not a purely single cell because the resolution problem, not only the resolution, but because of the way you can sample the tissue, there's no way you can only pick one cell under this probe. You always have the probe will penetrate maybe multiple cell types. So each spot you measured here is your readout of the matrix here. Each spot you have one expression level. But that expression level is from all the cell type under this probe. So here may have macrophages, some endothelium cell, or maybe you penetrate into cancer cell at the same time. So you have to do the deconvolution in order to know under this particular probe how many different cell types are there, how much each contribute to the expression level of those total rates. Yes?
Will you go back 2 slides? Like one more. So this package, it's specifically for bulk RNA-seq, but does it account for transcriptional bursting?
No, I don't think so. Yeah. See, this probably just use the marker gene database, which you have the information of the different marker genes for different cell types. They probably have a lot. Okay. So here we only have 2 as an example. So those are the algorithms behind those single-cell spatial transcriptome packages. They have this cell type marker genes database. Okay, so you know cell type 3 only G and D expressed. Then you have those under each of these probe, different cell type combinations. You do these 2 information, 2 matrices to help you to dissociate or deconvolute and give you the percentage of a different cell type. And then you can, so this is spatial transcriptome information, you can see, okay, the cell type 1 located mostly in this up right corner, and then in the middle of this tissue, most of cells are purple. To the end of this corner here, and there's more green cells in here. Okay, you start to see the change of the cell type concentration. Yeah, with that spatial information. Same idea, okay? They call them meta-genes. So maybe here is, instead of a individual cell type, they also learn the program of the genes getting involved in. Let's say same idea, okay? Okay, I think it's time for 5 minutes break, okay? Okay, so if you have a squared matrix A, and you can do eigen decomposition. Okay, here we don't have squared matrix because you cannot guarantee you have the same number of genes, same number of cells. But you can convert that into one of the opposition. You can transpose X and times X again. So this will guarantee you have a squared matrix. This will give you a gene by gene matrix. G by G, okay? Or if you do X, you can do X times X transpose. So here we'll give you a C by C matrix. So this matrix tells you the covariance or relationship or similarity between cells. And this matrix tells you about the gene similarity. So either by rows or by columns. So if you do the transformation, very easy, okay? So you transpose the matrix times itself. Depends on the direction. You either have a cell by cell or gene by gene squared matrix. With squared matrix A, then you can find, especially if this is a symmetric. Symmetric means the values in the off diagonal, if the same corresponding location, Aij will be the same Aji, okay? Then it will be mathematically guarantee you can have a eigenvector and eigenvalue. It's a real number. Otherwise, you can write into a complex number. But if you have a symmetric A matrix, then you can have some real numbers here. So the definition of the eigenvector, eigenvalues is the eigenvector. So this V is eigenvector times A is the same as a lambda. Eigenvalue is a constant times V itself. So most of the time you have more than one vectors you can discover. So you can put these two vectors together, become an eigen matrix, or if you have a two and this become two columns, and then corresponding eigenvalues will have two values in there. You can write them together, just like you write something together rather than you separate them. This is an example. This eigenvector for this with the corresponding eigenvalues is 6. So if you plug those numbers in, this A multiplied by this vector, okay, gave you this value here. This vector is the same as your eigenvector times 6, the same, right? So that establishes your relationship left and right. And from a previous lecture, you know the geometric interpretation of those vectors. Those vectors reflect the properties of A as a rotation matrix, whether it will change the direction of a vector, a data point, or not. And if you can find an eigenvector that is a data point that A will not change the position, the rotation position of V. So only make it either longer or shorter magnitude. So that's what the example you have seen many times. All right? Yeah, I should make this better resolution, but I just take this YouTube screenshot. I couldn't even find where the YouTube is, this video. Anyway, so when we do this square matrix A decomposition, this is also called matrix diagonalization. What you have to do is to rearrange this equation, okay, the definition equation. Put the V to the other side. So this is definition equation. So you have to move this V from the left to the right. Just you put a divide something, okay, you have to put this inverse of this matrix, okay, V here. In the middle of this diagonal. So if you have lambda, remember you have normally you have eigenvalues. If you have two eigenvalues, say 2 and 4. So when you opposite this diag lambda, this will put this 2 here, 4 here, and put a 0 here. Okay, so this function just transform the vector expression into a matrix. Why you want to do this, want to make this become a squared matrix? Because you can do this multiplication easily with other matrices. So that's why when you have more than one eigenvalues, you put in this diagonal form. And then you can multiplicate this eigenvalue, eigenvector. Okay, if you have multiple eigenvectors, this become a V matrix. So this form is you decompose, you do the eigen decomposition, and the so-called diagonalized the matrix A. This is the same thing. Remember here, it become not a negative 1. Negative 1 means inverse of the matrix. Well, this is easy, the transpose. So if you compute a transpose, a matrix, it just turn around the matrix. But this inverse of this matrix is more difficult to compute. But you can do this diagonalization if you have a real symmetric matrix A. Just like in this case, we have a real symmetric matrix. Then you can easily to replace inverse of the Q, okay, the eigenvector matrix. Rather than use the inverse, you just transpose it. This relationship still hold. So that's an eigen decomposition because this Q inverse and the transpose are the same. So why we need this, and there are some of those numeric lineage algebra and require this property in order to compute, such as matrix exponential is much quicker in this way after you decompose that, diagonalize the matrix. For instance, you want to compute A times A itself, so this is exponential. That's the power. Then you can diagonalize the first A into this way and times itself, right? And then you can rearrange, everything canceled out. At the end, you just have a D, which is the diagonal matrix squared. So this is very easy to compute. Then you just, after you compute this costs nothing because this just squared those two values. It's 416. Then you just multiplicate P and P inverse back, and you compute the square root of A matrix. Think about this 2 is a small example, but if you have a big number, so 25, then compute will be difficult. Apply to the matrix 25, multiply together if you compute in that way. But if you do this eigen decomposition first, then you only need to compute the diagonalized eigenvalues matrix, and no matter how much the power you take, 25 or even higher, is easy to compute. Then you just multiply it by back to this. So make this much quick. And in the matrix exponential is even harder. So you have exponential function and use this a matrix as a term. You can decompose that and change to this way, okay? To compute the exponent of the D matrix again, so this will be much easier. Okay, we jump to another decomposition method. This is 1976. So this old gentleman here is the inventor of MATLAB. His name is Professor Clover Moeller, and he's the founder of the MathWorks. Okay. So this is about SVD and its significance. Watch on YouTube. Yeah, I cannot make this sound work. Yeah, I just realized. Thank you.
To a mathematician, a matrix is a table of numbers which describes the relationships between the components of various kinds of systems. The system might be a bridge, an electrical circuit, a nuclear power reactor, or a sociological group. Each of the components of the system, the bridge struts, the resistors and capacitors, the neutrons, or the individual people, interact with the other components. The matrix elements represent the intensities of all these interactions. Analysis of the system using matrix techniques allows one to predict the effect of individual interactions on the overall system. Oscillations may be generated. Matrix analysis can describe the frequency and magnitude of such oscillations. This bridge near Tacoma, Washington, was designed and built before the effect of such interactions could be completely analyzed. The wind has reinforced some undesirable oscillations with unfortunate results. Modern computers are particularly well suited to solving problems involving large matrices. The designer of a system can obtain accurate predictions about the reaction of his system to external forces. The computer is used to simplify the matrix so that most of the entries will be zeros. One method is known as the singular value decomposition. The original matrix is represented as the product of 3 matrices. In the second of these matrices, the only non-zero elements lie on the diagonal. These elements, the singular values of the matrix, determine the frequencies of oscillation of the original system. The other 2 matrices must preserve the properties of the original matrix, so they are orthogonal matrices. Each is the product of elementary matrices, which affect only 2 rows or columns. Scientists at Los Alamos Scientific Laboratory have been studying methods for the accurate and efficient calculation of the singular value decomposition and of describing to computer users how the computation is carried out. In this 3-dimensional representation of a small matrix, each pyramid depicts the magnitude of a single matrix element. The diagonal of the matrix runs from the upper left to the lower right. Orthogonal transformations will be used to introduce zeros into the matrix. The initial transformations put zeros into the first row and then the first column. Each transformation affects 2 other columns or rows shown here in blue. Later transformations introduce zeros into later rows and columns, while preserving the zeros already obtained. It is not possible to annihilate all the off-diagonal elements in a column, so this first stage produces a matrix with non-zero elements directly below the diagonal, as well as those on the diagonal. This film is produced by exposing a single frame each time a matrix element is altered, to illustrate roughly the computer time actually required for various phases of the computation. The elements shown in red are involved in auxiliary calculations which require time, but which do not directly alter any elements. As more zeros are introduced, the rows and columns become shorter, and each transformation requires less time. The second stage of the calculation is a repetitive or iterative process which reduces the size of the remaining off-diagonal elements, but does not zero them in a single step. During the process, new elements, shown in red, are temporarily introduced into the matrix. The calculations now being shown are primarily intended to reduce the size of the off-diagonal element at the lower right-hand corner. Once an off-diagonal element has reached a negligible magnitude, it is set to 0. The computation of the corresponding diagonal element is then complete, and the resulting singular value is shown in blue. During these iterations, most of the other off-diagonal elements are also being reduced in size, although not as rapidly. A few of them will occasionally increase in size. This makes theoretical analysis of the method difficult, but does not seriously impair its overall effectiveness. Since elements are being depicted according to logarithmic scale, the off-diagonal elements we see are actually quite small by this time. The transformations are still changing the diagonal elements, but the changes are not visible on this scale. Once a few of the singular values have been obtained, the off-diagonal elements have become so small that each of the remaining singular values is obtained with a single iteration. The final diagonal matrix, together with the 2 transforming matrices which produced it, then provide the basis for a complete analysis of the original matrix and the system which it models.
Yeah, so this is just an example application of the SVD in engineering.
SVD, singular value decomposition, is a grand finale of linear algebra. On one hand, it combines all important concepts of the subject into just one theorem. On the other hand, it's extremely relevant and applicable in the age of data science and machine learning. This is a
Yeah, this is too long, and 15 minutes, otherwise I will keep playing. Anyway, so this example was try to learn this diagonal matrix because that's important to tell the direction of this engineering problem, the shift. So they have defined another two matrices already in the system. So, but in general, this is a very, very important decomposition method because it's applied to any shapes of this matrix, not required to be squared matrix. So they're much wider range of applications. You can decompose basically every kind of matrix. So it works like this. So you decompose a matrix A into three matrices, U and V and the sigma in the middle. So this is an example. You decompose A, so this is not squared matrix, it's 2 by 3. And you make that U matrix here, and this is a sigma matrix. The position only got values in the diagonal position. So this is a diagonal matrix. So those are the scaling factors for different components. And then you have this V transposed version of this V matrix. So this forms the SVD. So the values in this diagonal position matrix is called singular values. So those are indication of the magnitude of the matrix contribution of the components in the U and different components. Okay. So that's the writing. Okay. So the A matrix is M by N, and then you have M matrix here, and N by N matrix here. In the middle, you have this matrix M by N. All right. So in this example, M is smaller than N, so this diagonal matrix only have values in this top two and M position, but the last column are all zeros. As we said, A can be considered as a rotation matrix. So once you decompose A into three components, the first component is a matrix, squared matrix, can be considered a rotation matrix. The second one is not squared, but you can truncate the last part of zeros becomes, but this is a scaling factors. And then U, another squared matrix, can be considered as a rotation matrix. So SVD actually decompose this matrix A into these three parts. If you consider this V and U as a rotation matrix, then you can think about apply this rotation matrix to two data points. Okay, in this case we have two data points, blue and red. So the first rotation matrix will rotate we'll rotate this data points, okay? We'll apply V. So this is just an illustration to rotate some angle. And then the information contains in this sigma matrix is the scaling factor to make different components either longer or shorter. So you actually stretch the magnitude of your data, but they based on the direction. And then you have another rotation matrix, and then you rotate data point again. So you start from the data in this blue, red direction. At the end, you change it and then also stretched. So the matrix A will be described as an operator contains both rotation and scaling. And you decompose the A into three matrices and use three matrices to describe the property of A if you consider A as an operator. And this video has been viewed many, many times. It's very good explanation of SVD by this professor in Washington University, Seattle. So if you have time, I hope you can watch that.
This week, we're changing
40
topics
minutes.
to...
Very good talk. Let's look at examples. We decompose this matrix into U sigma V, and each have some meaning of those components. Those are the documentation. I believe those are the books from computer science, from a medical doctor, medicine, and then you have frequency of different keywords appeared in those books. So in computer science book, those you have more data information retrieval. Well, in the medical books, you have brain, lung, so that's the frequency of those keywords appeared. But if you don't know those information about the categories, you can decompose this matrix into these three, okay? And then this matrix is documentation to the concept. You have two different concepts, like K components. And this algorithm will decompose this into something like this, and those concepts with terminologies. So there are two concepts, and each associated with different keywords, the frequency or strength of association. Okay, you decompose that into sample and the concepts and the keywords, and in the middle you have concept strings. So this helps you to learn from this original matrix of the potential concept embedded in the frequency of this keywords appearance in this sample matrix. Another application is to denoise of SVD. If we look at this example, 1, 2, 3, 4, 5, 6, A was decomposed into this, and that. And we look at the strength, the magnitude level of the second component. So this is much smaller than the first one. So what that means is the contribution of the second component to reconstruct A is much smaller than the first one. So what we can do is to, in some applications, you can delete those columns. You can delete this, only use this, that, and this. Use this, use that. To approximate this. Because you learned from a single value vector, and the second component is much less important than the first one to explain all the variability in original. So that's why you can truncate and only use top ones to reconstruct. So this is a small example. You have two components, you get rid of one. But if you have large applications like this, you decompose and have SVD, have many genes. You can say, okay, I don't need those less important genes. I only need the top 50 to reconstruct my original expression matrix. But same idea here, okay? So this indicates the truncation of the application. So you decompose the original matrix A into m by m, n by n, but you only keep those important ones, components, and this only keep top P, okay? And get rid of the rest of them. So you don't need this part, you don't need this part, you don't need those part. You only select top P components to reconstruct the original matrix A. You will find, you will have very good approximation if you select those top ones, because the rest of them are just noise, random noise. This P can be much smaller than M. If you have a number of genes, so M genes, 6,000 by 6,000, then you only need top 50. So this P number is much smaller than 6,000. You keep top 50 genes, components in there. So as I write in this matrix form here, and you can separate into columns, components. But this example is for non-negative factorization without this middle term sigma here. But the same idea here, okay? You have U and V contains two components. That's why you have two columns here and two rows here and two non-zero values in the sigma diagonal matrix. And you can write in this vector-based multiplication way. Just do this, okay? This vector times this value will create the first component's contributed values. And this one, so this A1 plus this A2 gave you original A approximation. You can separate them into more if you want to have a better estimation, okay? But those may be not important, and that's why you can delete them. Okay, those components. So this is the most important concept I want you to learn, to know, and put that into your mind. Then you can decompose a matrix into multiple components and select the most important ones, to use the most important ones to reconstruct your original. This reconstruction lost the function, lost information, and because you never reach to the because that's only optimization, that's only approximation. But there's value is you already learned the most important components, and less important ones have been deleted. So this is a denoise procedure to make your data sometimes cleaner to as for the downstream. Every major numerical computing package should have SVD function, okay? In SVD, you just call, you give the matrix, as I said, any matrix. It will return 3 U matrix, S vector could be the matrix after diagonalization, and the V matrix. So look into some details, how this SVD was computed. So inside SVD function, it's actually doing this. It's doing this, create C by C matrix and a G by G matrix, and then each of them process. So the eigenvalue decomposition is inside the SVD function. So this is called the indirect method. So you do a matrix, okay, non-symmetric, but after that, you do the single eigen decomposition, and you do the same for A times A transpose. So here you learn the V matrix, here you learn the U matrix, and then you put them together. One of the applications of SVD is to help you to compute the inverse of the matrix. So this procedure is called a pseudo inversion, okay, PINV. Direct compute of inversion of the matrix may be difficult, so that's why you first do the SVD to decompose your input X matrix into USV. And then you inverse the middle S matrix. That's easy because this is a diagonal matrix. So after you inverse this, you multiplicate those two back, and this becomes the inversion of X. Okay. So this SVD help you to compute the inverse of matrix if the original inversion is difficult. So that's why where you can use the inversion of matrix, you can do the linear regression, remember? If you want to use a beta matrix, a BE times X. So this is a linear regression, and you want to learn beta, then you can compute the beta as inversion of X times Y. Inversion is difficult to compute, then you use pseudo inversion, and inside the pseudo inversion you actually call SVD to decompose this, make this inversion become the problem to inverse the square, the S matrix. That's much easier than compute the whole thing. So then you construct it back. So there's some use of this. At the end, I will mention this called independent components analysis. So this is application quite a lot in the signaling processing procedure. So just imagine you have a mixed signals. And a good example is to solve this cocktail party problem. The setup is like this. You have, you are in a cocktail party, then there are some background music noise, and also the human conversation voice. So those two sources of the information was mixed, and then you prepared two microphones at different locations. One is close to the background music, another microphone is close to the human conversation. Then each microphone will sample the source of information, the sound information, but in a slightly different proportion. See, this microphone take 90% of background music and 10% of the conversation. Well, this one will take 20% background music, 80% of conversation. And then the sound will be mixed and sampled. You sampled a mixed information, but in a different proportion. So once you have these two inputs and gave to the ICA algorithm, ICA will reconstruct the human voice and the background music for you, okay, to separate them. And we can play with some examples. It's really, really cool idea. So the formula behind this ICA analysis is you want to reconstruct a matrix A. So this A will tells you the different proportion of the combinations of two sources. Okay, you have two samples, sampling. You have two microphones. That's why you have a 2 by 2. Okay, so it will reconstruct A to help you to transform your S, the input signal, and give you the decomposed signal vectors or matrix, depends on your input. And this procedure is try to approximate this mixture. So this is like mixture, the way how you combine, as I said, 10%, 90%, 80%, 20. So this A is the way you think you mix the signals mixed. So you want to learn this A. There are different applications and implementations, and we can look at this once we have our computer session and look at those examples. So the algorithm, when you open up, start to take the input mixtured signals and meaning and normalize and add this whitening.
another statistic procedures. The purpose of this is to help the algorithm converge much quicker after you do this whitening steps. And inside here, there's updates of the different parameters in the A matrix. Then at the end, you will have a separated information signal. So application for this, including EEG, okay, so signal from a different location of the surface of your brain, then recorded, called the different channels. And those channels, each sampling points contains information from multiple locations of your head, right? This is a mixture, depends on the distance at which channel take what source of the signal. And then you try to reconstruct those components and learn from the different kinds of wave or different signals. Yeah, so that's all. So we probably should find some single cell data analysis examples, but I haven't got any examples for the ICA applications. But it's there, and we probably should consider, especially if we have time series information from a pseudo time or different signals. We can record it from individual cells, but that's something with potential applications. If you found any good articles related to the ICA applications of single cell data, I would like to know. All right. Yep, thank you, and see you Thursday.